Good afternoon and welcome to the New South Wales Stem Cell Network webinar on exosomes and regenerative medicine. This session is being recorded. All participants will be muted, but we encourage you to use the chat and Q&A functions throughout the afternoon. Welcome, my name is Janet McPherson. I'm currently the product specialist for the cell and gene therapy portfolio of Cytiva across Australia and New Zealand. I'd like to take this opportunity to thank our sponsors, Sartorius, Thermo Fisher Scientific, Cytiva, the Paul Corporation, and Bard One Life Sciences Limited. Their contributions help support the network in the provision of educational content. If you have not already done so, please take a moment to review the services offered by each of the sponsors and follow up with them directly. A Dropbox link was also shared to contact that might be of interest to you. Since its establishment in 2002, the New South Wales Stem Cell Network has organised over 30 events, including in-person workshops, educational programs, and more recently, webinars. The workshops have covered a broad range of topics related to scientific, clinical, regulatory, and ethical aspects of the use of human cells for research and the development of therapeutics, particularly for regenerative medicine. I'm delighted to have been involved with the network for almost 15 years supporting these educational programs and workshops. Today's topic of exosomes is an emerging area of interest in regenerative medicine, and it was suggested by Dr. Daniela Goldberg, one of the founders of the New South Wales Stem Cell Network. Our speakers today are Associate Professor Lim, who's a group leader at the Hudson Institute of Medical Research, and she'll speak on cell-free regenerative medicine for perinatal applications. Then we have Dr. Sai Kang Lin, who's a research director at the Institute of Molecular and Cell Biology at A-STAR in Singapore, and she'll talk on exosomes in the treatment of psoriasis. And their talks will be followed by Professor Andy Hill, who's a laboratory head at the La Trobe Institute for Molecular Science at La Trobe University, on standardization efforts for EV research and applications in neurodegenerative diseases. All three of our speakers are very active in the professional societies, the International Society for Extracellular Vesicles and the International Society for Cell and Gene Therapy. We're limited today by time, but we know that there are other organisations working in this field to develop novel treatment options. We'll be taking questions for each speaker after their presentation, and then the group will meet at the end for a panel discussion. Please do feel free to enter your questions in the Q&A window at any time. If you would like a specific speaker to address your question, then please include their name. It's my great pleasure now to introduce Dr. Rebecca Lim from the Hudson Institute to give us an introduction into the work that she's been doing over many years to help treat babies with inflammatory lung disease. Over to you, Rebecca.
Thanks, Janet. I'll attempt to share my screen once that's stopped.
I need to change that, I think, sorry.
All right, can you see my screen okay?
Yes, thanks, Rebecca.
Perfect, thank you. So thank you very much for the kind introduction, Janet, and it's a pleasure to be speaking to you guys today. I am at the Ritchie Centre, Hudson Institute of Medical Research, and also have an adjunct appointment with Monash University. I'll be speaking to you today about some of the work that we've been doing over in our lab and in our department. The Ritchie Centre is a research arm of the Department of Obstetrics and Gynecology, as well as the Department of Pediatrics. So perhaps it's not that surprising with that in mind that our first-in-human trials with cell-based therapies have been in neonates with incredibly unwell neonates with chronic lung disease. And specifically, the disease that we're looking at is called bronchopulmonary dysplasia, or BPD for short. It's a chronic lung disease that commonly affects extremely premature infants, essentially infants that cannot breathe on their own, require respiratory support, ventilatory support, require antenatal steroids, for example. Those are essentially the patient population that we've been focusing on. If you look at lung development in humans at the stages where we're talking about this extremely premature stage, their lungs are at this canalicular stage and at sometimes the saccular stage, where these infants are born with, by extremely premature, we mean less than 28 weeks of gestation. So these extremely premature lungs are kind of forced to develop a little bit sooner than would have been previously planned through the use of steroids. And the addition of steroids, you kind of force that differentiation into alveoli that can then perform gas exchange, albeit not adequately. So then these infants require oxygen support and sometimes ventilatory support if they cannot actually ventilate on their own. So this combination of complex factors results in this very simplified alveoli. And by this, I mean lungs that look like this, where you've got lots of air spaces and there's not a lot of tissue to surface area for gas exchange, and this vascular alveolar mismatch. A combination of all of these factors are essentially what gives rise to the clinical condition known as BPD, bronchopulmonary dysplasia. What's interesting as well and noteworthy is that these infants are also prone to developing secondary complications such as pulmonary hypertension. As a consequence of that, right ventricular hypertrophy and cerebral palsy. So our team has been looking at a cell type known as amniotic epithelial cells. So these are the cells that are derived from the amnion, and the amnion is one of two fetal membranes that surround the fetus during birth. So they're not stem cells, they are, stem-like cells perhaps, but they are definitely terminally differentiated adult cells, adult type cells. The HACs for short, they do bear markers of pluripotency. I've just listed them over here. They're probably quite familiar to this particular audience. However, as mentioned before, they are epithelial cells, so they are EpCAM positive. They're heterogeneous. If you try to culture them, they don't culture terribly well. But if you do try and culture them, what you will notice is there's an adherent population, a loosely adherent population, and a non-adherent population. And that heterogeneity carries through to the cell surface markers as well, if you attempt to characterize them that way. The amnion perhaps is not so unusual when it comes to regenerative medicine because it has been used for wound healing, and that goes back for well on to a century now. The first documentation of the use of the amniotic membrane actually dates back to 1910 when it was used as a surgical bandage, but I guess contemporarily it is used for things like biological bandages for burns, for skin grafts, not as a skin graft, but as a bandage for skin grafts. It's used as a scaffold for culturing limbal stem cells, for instance, and in this case, ophthalmology has continued to use it throughout those decades. What's important to understand is that the amniotic membrane, along with the rest of the placenta, actually has quite an unusual immunomodulatory characteristic. If you look at the way the human placenta has evolved to develop, placentation as a process in pregnancy is actually quite invasive in humans. So you get fetal antigens that are constantly being presented to the maternal immune system as that invasion occurs throughout the course of pregnancy, which means that placental tissues or perinatal tissues that are derived at the end of a gestational period from a healthy, uncomplicated pregnancy has actually been able to tune the mum's immune system quite significantly. And we think this is one of the reasons why they're tolerated allogeneically incredibly well. We also do know that failure to adapt to pregnancy, so when this process doesn't occur and the fetus isn't carried to full term, you get massive devastating outcomes associated with that. So going back to BPD and bringing those two aspects together, we're looking at a situation that is highly inflammatory, where there is a significant amount of cell death, there is deficient alveolar polarization and vascularization. In, I guess, old BPD or very injurious ventilation strategies, you can also get barotrauma that often leads to fibrosis within the lungs, although this is less common these days with gentler ventilation strategies. But all in all, this is a very complex disease with requiring a multifactorial sort of therapy. And if you pull in what I just mentioned earlier on about what the HAECs do, what we do find and what we have reported previously as HAECs in preclinical settings are immunomodulatory. They can activate endogenous stem progenitor cells within the lungs to overcome that alveolar simplification that I showed you at the start of the talk. They are pro-angiogenic. And this is noteworthy because angiogenesis and alveologenesis go hand in hand during lung development. And they are also cytoprotective, potentially saving some of these cells from cell death within the lungs and reduced fibrosis in those animals. So what's also worth mentioning right now is that some of this work that we've done with the cells also involved the preterm infant center of research excellence that's been led by Dr. Jane Pillow here, who's a neonatologist. And so we've used a combination of small and large animal models to take us to our first in human trial. And this is just a summary of some work that was done earlier. This is first in human trial with 6 infants. The first 3 were administered cells intravenously after having had severe BPD and being ventilated. So they were intubated. The next 3 in this cohort of 6 also had severe BPD, but these babies were on CPAP or high flow oxygen. And this work has now, it's gone quite a number of years now, and we have the 2-year follow-up that's been reported already. What might be worth noting as well is that, as I mentioned earlier on, you don't get babies with just severe BPD. They almost always end up with secondary complications. Pulmonary arterial hypertension is one of the very common ones. So the 2-year outcome has been reported already. So if you'd like to have a look at that, that might be interesting for some of you who are interested in going into neonatal regen med for neonates rather. This is baby Flynn, and this was him shortly after receiving the cells, and 2 years plus on, he's doing very well. And this then, I guess, gave us the impetus to go to a dose escalation trial, because this first in human study was with a very low dose of cells. It was 1 million cells per kilogram, and only in infants that already had established BPD. So now in our current trial with the dose escalation study, these are infants where the intervention is a lot earlier on, and they are typically recruited within 14 days of birth. They are still extremely premature, and we're looking at it from a more prophylactic sort of approach. So infants that are at high risk of developing severe BPD and its associated complications are then recruited to this trial. The current challenges, however, and I guess our chair here has a very unique insight into this, these neonates are extremely fragile. So the challenges that we have implementing a cell-based therapy is that you have very narrow and hyperactive pulmonary capillary bed. So the risk of pulmonary emboli is there. Slow infusion is required for these cells because these infants have incredibly small blood vessels, and if you were to perform an IV infusion at the rate that you do with adults, you could cause some fairly serious side effects. The cells, because of the slow infusion, cells will settle in the lines. The HAECs are a fair bit bigger than PBMCs, for instance. So while you don't really have to worry about this when you're doing a blood transfusion, for example, certainly infusing the HAECs, we do see them settling in the IV lines. There are very limited CMV negative donors, and of course these infants cannot, we cannot simply expose them to that kind of risk. So that gives us a very limited number of donors that we can go to. HAECs expand very poorly, which means that you have a new donor and new batches of cells that have to be tested and then released into the trial. So we continually rely on primary cell lines. And of course, along with that, we have donor variability. So there are quite a few challenges that kind of led us to think, can we do better? And this is where I guess our team personally, this is what led us to thinking about using EVs or extracellular vesicles. So In this case, what I want to show you right here is one of the very early bits of work. So we started collecting this data probably in 2014. So these methodologies are no longer really relevant, probably superseded, it's probably the appropriate word to use here. But here it is, nanoparticle tracking analysis showing us that the EVs kind of accumulate around the 100 nanometer size and diameter. They do have not cell surface, but I guess surface markers that are typical of EVs, these tetraspanins over here, CD9, CD81. We were quite keen to kind of align ourselves with the minimal requirements in the past that have been published by the International Society for Extracellular Vesicles. So based on those ICEV guidelines, we then kind of looked at ALX, GRP94, and cytochrome C. This is comparing the EV protein levels to HAC levels. And what was quite interesting is that HACs that express a lot of this immunomodulatory HLA-G protein, EVs from those cells also package quite a fair bit of it. Scanning electron microscopy over here showing you that cup-shaped morphology that used to be seen as one of the criteria for so-called exosomes, but possibly not the case anymore. But because of that, at the time, we were quite keen to see if we could observe those multivesicular bodies and then binding onto the plasma membrane for release. So these were those guidelines or minimal requirements that I spoke about. This was, I think, a 2015 paper. This has since been superseded. So those of you who are new to the field or are keen to align your research more closely with the MyCEF guidelines, there's that paper there for you. So a bit of a plug for our paper. But personally, we were interested to see what the effect was on the yield and potency when it came to different donors. So we went ahead and looked at term donors and preterm donors. So going back to what do we use for a clinical trial if we were to use EVs? We did a mass spec to see how much overlap there was from the cargo of derived from EVs from preterm donors compared to term donors. Turns out not so much. There's also not so much of an overlap with the cargo from EVs derived from MSCs. However, the yield was kind of comparable. So preterm EVs, if you correct it for the number of cells releasing the EVs, they were kind of similar. However, they were more immunosuppressive, if you like, so they could reduce T cell proliferation quite a fair bit more. And in terms of their ability to express CD133, so this was, we had actually subjected the EVs to testing with I think it's a 37-plex sold by Miltenyi. So looking at 37 different markers, and CD133 was one of the ones that was significantly changed. And preterm EVs actually display a lot less CD133 on their surface. And this was important to us because if you look at the role of prominent 1 or CD133 is its role in angiogenesis via Wnt signaling and the expression of VEGF-A, which of course is an important ligand as well. So with that in mind, we went ahead into a preclinical model of BPD and we tested them with either EVs from either term or preterm HACs. And just a quick run through of this model, at E16 of gestation, the uterine horn is exteriorized and each one of these fetuses receives an LPS injection in utero, intraamniotically. They're then returned into the mum and allowed to litter down their pups. And once they are born, they're exposed to 65% oxygen. And then at day 4, we perform an intervention and we call them at day 14 or week 6, corresponding to sort of early, I guess, childhood and adolescence or early adulthood at this point. So looking at alveolar simplification, if you even just ignore the quantitative data, what you'll notice is that this injury group starts to look a lot like the human BPD, where you have simplified alveoli. So the animal model kind of holds true in that sense compared to the healthy animals. And this is what we previously reported with the HACs that we were giving. And this is what it looks like with the term compared to preterm. So it looks like preterm doesn't quite perform as well as the term ones, because you can see over here, you do get some more complex alveolar structures in here. And if you were to quantify it by tissue to airspace ratio, you see that difference where you get a drop in the tissue to airspace ratio. In the injury group, you get a restoration with the HACs, and more comparable to that is the term if you compare that against the preterm. Previously, we had reported on the effects on bronchoalveolar stem cells, and I had mentioned that the HACs did previously report that they could activate endogenous stem progenitor cells. So we looked at the bronchoalveolar stem cells, and those are the stem cells that sit at the terminal bronchi, and the type 2 alveolar cells, which are commonly known also as progenitors of type 1 alveolar cells. So just a quick reminder that the type 1 cells are the ones that are responsible for gas exchange. So it looks like it doesn't matter if you give them EVs from term or preterm HACs, neither of them really activate the stem progenitor cells, but the term EVs did activate the type 2 alveolar cells. So it may very well be that because of the enrichment of different cargo, or the fact that the EVs are not able to respond to the environment real time like a cell would be able to do, that could be why we're getting these differential effects, but that's a working hypothesis we have at the moment. Bronchopulmonary dysplasia is commonly leads to secondary complications like pulmonary arterial hypertension, as I mentioned. So we were quite keen to see if we could see those muscularization, and lo and behold, we see this thickening. So this is alpha smooth muscle staining. So this red stain here is for those smooth muscles surrounding the smaller blood vessels in the lungs. And with the HACs, we saw that there was less of that thickening. The term EVs, there was, somewhere in between, if you like, and the preterm, the EVs from the preterm HACs didn't seem to do as well. And this is what we quantified essentially. Preterm EVs just didn't do quite as well as the term EVs. So in summary, the prematurity of the donor didn't seem to affect the EV yield, but it did have some significant implications on the potency. So we're thinking if we were to move ahead to clinical trials, we would look at term donors. However, there are a number of different questions that we are asking, such as, having better angiogenesis may be better for the lungs, but in neonates who are on high oxygen, there is a risk of worsening retinopathy of prematurity. So that is a risk that we would look at. And we've been delivering the cells IV, but you could deliver the EVs intranasally. But there are pros and cons to that, because crossing the blood-brain barrier will allow us to address brain injury that's happening at the same time, such as hypoxic ischemic encephalopathy or white matter injury that's been associated with cerebral palsy. But could this be deemed to risky in preterm neonates? There are heterogeneity of EVs, and ensuring consistency between batches will be an ongoing question that we'll attempt to address. And what I mean by that is this is an image captured on technology that basically is chip-based, and each one of these colors corresponds to a tetraspanin. So red, green, and blue. Red, green, blue. And we have a number of different tetraspanins associated with the different colors. And you can see, even without going into too much detail, that you've got a lot of colors here. It's almost like Christmas lights. So that goes to show that the EVs that you get from one single donor, they're not all the same. So how do we ensure that if you have this heterogeneity, that you have the same level of heterogeneity between donors and between batches? So that will be a challenge that I think the field needs to address if we want to proceed this way. And of course, what other aspects of donor health will influence the quality of the EVs? So I'll leave it there. Thank you very much for your time. I won't go through all of these people, but I do want to thank Cytiva and Thermo Fisher, who have been our industry partners for this project. Thanks.
Thank you, Associate Professor Lim, for a very interesting talk. And I always have find it difficult each time I see the images of the very premature babies, and how amazing it is that the team of neonatologists can help bring these children forward in life, and so that they can survive. It's great. It's really exciting. There's one question that's come through on the Q&A, and I believe that you've addressed it in part already, but Professor Kaldup Sidhu has asked whether you've carried out any preclinical work with the amniotic cells before you started the clinical trials, and also before you go into your amniotic derived EVs, will you be doing more work?
Yeah, so with the AECs, before going to clinical trials, I think we're sitting about 12 years worth of preclinical data. So we didn't move into humans lightly or frivolously. The amount of hours and I guess NHMRC research dollars was quite significant. And I expect to do the same, that we will do the same with the EVs. This is an extremely protected patient population, and we wouldn't expect anything less.
And then there's a follow-up question also from Kaldup asking whether you would consider using iPS-derived EVs to address your heterogeneity issues.
Certainly we would consider that, but I don't think it addresses the heterogeneity issues. I think Sai-Kiang and Andy may be able to address this, or maybe addressing it in their talks as well. But even from a single cell line, from a very what appears to be a homogeneous clonally derived cell lines, you get heterogeneity. And the heterogeneity may be the secret in the sauce. Like, let's not be too reductionist about it either.
Thank you. And we've just got time for one last question before we move on, and that is, can the EVs be delivered through inhalers?
Absolutely, and there've been a number of very clever people who've been working on this. The issue with inhalers tends to be that what you're trying to target are the distal airways. Inhalers tend to result in drugs, any kind of drug, being caught up in the upper airways and in the nasal tract. So you do get diminishing returns as the target organ you're going is further down.
Thank you very much, Rebecca. And just thank you for your talk.
It's a
bit of an unusual situation when we're in the virtual environment, because we can't use our normal clapping to thank our speakers, but I'm sure you're all joining with me to say thank you. It's now time to turn to our second speaker for this afternoon, and that's Dr. Sai-Kiang Lim, who is based at the A-Star Institute in Singapore, and she has a very extensive career in analysing and characterising extracellular vesicles. I'm hoping that she'll also touch a little bit on what the difference between an extracellular vesicle and an exosome might be. And I'll hand over now to Sai-Kiang to present. Thank you.
Well, thank you very much. First of all, I'd like to thank the organizer, the New South Wales Stem Cell Network, Janet and Bernie, for this opportunity to speak and share some of my work here. So I'll be talking about MSC exosomes in the treatment of psoriasis. Now, before I start, this is my disclosure. I'll start briefly with MSC exosomes. So MSC exosomes, as Rebecca has described, is extracellular vesicles. MSC exosomes, of course, is derived from mesenchymal stromal cells called MSC. So it's a widely used cell type. In fact, the most widely used cell type in regenerative medicine research and also clinical testing. Its use has been always predicated on its ability to, it's thought to, well, it's predicated on the hypothesis that upon transplantation, the cells will home and to the injured tissue and graft and differentiate to form new tissues. However, in many animal studies as well as clinical trials, it's been observed that even in the presence of functional improvement, there's very little evidence that the cells do engraft and differentiate. So in 2006, Arnold Kaplan proposed that these cells may access its effect through its secretion. And in 2009, Giovanni Camiosi group reported that the 80 nanometer to 1 micron microviscous are rhinoprotective. A couple of months later, in 2010, my lab reported that the 100 to 200 nanometer exosomes were cardioprotective. So both exosomes and microviscous are essentially extracellular vesicles. They are bilipid membrane vesicles. They have slightly different characteristics, but they also have very overlapping features. Now I'd like to sort of walk you through how we get to the discovery of cardioprotective MSC exosomes. In 2007, we reported that MSC could be derived from human AS cells. We also showed that these cells can be cultured in a serum-free media so that we could analyze the secretome. And we find that the secretome has very high therapeutic potential, and we tested them in a mouse and pig model of myocardial ischemia reperfusion injury. And we observed that the conditioned media from the MSC is cardioprotective, but not conditioned media for HEK cells. And we also observed that the active agent, the protective activity lies in the greater than 1,000 kilodalton fraction. We then spent the next couple of years looking for the active agent, and we isolated exosomes and found that they could also protect the heart to the same extent as the conditioned medium. Now, our characterization of the exosomes in up to 2010 was in compliance with most of the guidelines in the MySelf 2018. The MySelf 2018 is actually the ISEV position paper on best EV research practices. And by 2010, we have fulfilled most of the guidelines on how to characterize EV. And I believe Andrew here would be perhaps talking about the MySelf 20 in greater detail. So I will now skip. Now, after our first report that MSC exosomes was effective against myocardial reperfusion injury, my lab and many others around the world also find that MSC exosomes were effective against many diseases. In my lab, we observed that the MSC exosomes was effective against graft versus host disease, induced liver toxicity, osteochondral injury, acute radiation syndrome, and aging. So today I'd like to share with you our study to assess whether MSC exosomes are efficacious for psoriasis. Now, psoriasis is a chronic inflammatory disease and are highly tractable to immunotherapy. And we believe that exosomes could be therapeutic for psoriasis because we have observed that MSC exosomes can modulate the immune system. And this come from two areas. One is our studies of MSC exosomes in animal models of various diseases. Invariably, when we observe therapeutic efficacy, we also observe that the animals have significant level of immunomodulation. So for example, in cardiovascular reperfusion injury, the exosome not only reduce infarct size and increase cardiac function, it also reduces neutrophil and macrophage infiltration in the tissues and also the white blood cell count. Similarly, in graft versus host disease, in this model where we had 100% lethality, exosomes could rescue about 50% of the animals. And in these surviving animals, we see a significant increase in Tregs. In the osteochondral defect repair model, we observed that there was improved cartilage repair. And we also observed that in this repair tissue, there was increased infiltration of M2 macrophages and a decrease in M1 macrophages. We also observed that the amount of inflammatory cytokines IL-1 beta and TNF alpha was also decreased in the exosome treated animals. There's no effect on IL-6. In aging, we find using naturally aged mice and also the progeric mouse model, the aging symptoms were reduced by exosome treatment. We also observed that the senescence-associated secretory proteome factors also decreased similarly, and also IL-6 and IL-1 beta were also decreased in multiple tissues of the exosome-treated animals. So altogether, it suggests that when we administer MSC exosome in animal models, it could modulate the immune system, and this is associated with an improved therapeutic outcome. But independently, we also observe that the MSC exosome has many potent intrinsic immune properties. For example, MSC exosomes has a very significant level of toll-like receptor activity, receptor 4 activity. So we find that exosomes could activate toll-like receptor 4, but not toll-like receptor 2 in a HEK toll-like receptor reporter cell line. And we also find that this activation of toll-like receptor 4 is not sensitive to polymyxin B, suggesting that we are not looking at an LPS contaminant in this study. And we could attribute 75% of the toll-like receptor 4 activation to the EDA fibronectin that is highly present in our exosome preparation. And we could abolish this activation using neutralizing antibody. And finally, we observed that the toll-like receptor 4 activation by exosome is quite different from the LPS activated toll-like receptor 4, which leads to pro-inflammatory cytokine production. In toll-like receptor 4 activation by exosomes, it leads to a increased anti-inflammatory cytokine production, in this case IL-10. And you can see that for the pro-inflammatory cytokines, exosomes actually induce very low level compared to LPS. Now, we also observed that exosomes could induce Tregs in vitro. So if we incubate CD4 positive T cells with CD11C positive APC cells, in the presence of exosomes, we find that we could polarize the CD4 positive cells into Treg cells. Incidentally, the use of anti-CD3 and anti-CD28 could not replace this allogenic CD11C antigen presenting cells in this Treg induction. We also observed that MSC exosomes carry a very high level of CD59. Now, CD59 inhibits the last step in the complement activation cascade. So it inhibits the formation of the membrane attack complex, or the C5b9 complex. And we could assess the activity of CD59 in our exosomes using a sheep red blood cell lysis assay. In the presence of complements, the sheep blood cells will be lysed. But we find that in the presence of exosomes, this lysis is inhibited. And this inhibition by exosomes can be abolished using anti-CD59 antibodies. So in all, what we have observed is that therapeutic efficacy in animal models is associated with immune modulation. Exosomes has many intrinsic immune activities, and they are mainly anti-inflammatory. Therefore, there's the MSC exosome has potential to modulate the psoriatic immune pathology. Now, the immune pathology of psoriasis is very complex. It involves multiple cell types and cytokines in multiple crosstalks. However, the key consensus is that the IL-23, IL-17 axis is central to the psoriatic immune pathology. Now, consistent with this consensus is the fact, actually this is a fact, that the IL-23 and IL-17 therapeutics are very effective against intractable psoriasis. So a lot of the antibodies against IL-23 and IL-17 are now widely used in the clinics, and they are proving very effective against the intractable psoriasis. So the question that we ask then is, can exosomes alleviate psoriatic immune pathology? More specifically, we were interested in the question, can topical application of MSC exosome attenuate the activity of the IL-23, IL-17 axis in psoriasis. Now, to test this, we formulate the exosome in a cream so that we can apply them onto the skin. Now, for our experiment, what we did was to induce psoriasis using IMQ by applying an IMQ cream on the mouse skin. And after that, we apply exosomes for 7 days, and then we harvest the tissues. Now, what we observe is that when we apply IMQ on the skin, we cause a psoriatic skin phenotype. And we also induce systemic inflammation as evidenced by the enlarged spleen. So the spleen is enlarged about twice the normal size. Now, what we observe also, there's no difference in the skin phenotype and spleen enlargement between the exosome and the base cream treatment. However, when we look at the IL-23, IL-17 axis, what we found was that the IL-23 and the IL-17 was significantly reduced. So was C5b9, which, as if you can remember, it is the terminal complement activation complex. So we were actually quite taken aback by this observation because we were also doing looking at the dermal penetrance of exosomes when it is applied topically. What we observed was that when we apply covalently labeled fluorescent exosomes to human skin explant topically, we observed that the fluorescence remain localized to the stratum corneum. And at 24 hours, it still remains in the stratum corneum, but of course the fluorescence has much reduced. And when we look at the amount of exosomes of the fluorescent in the underlying culture medium, we've calculated that less than 1% of the fluorescent exit into the underlying culture. And what this means is that topically applied exosomes do not transit out of the stratum corneum. So this is a bit difficult for us to envisage how the exosomes can reduce inflammation, because as we know, the stratum corneum in our skin is the upper topmost layer in our skin, and it's made of dead conified cells. So how would the exosome attenuate immune activity in the psoriatic skin if it stays in the stratum corneum? Now, it is reported a long time ago that psoriatic skin or IMQ-treated skin has a lot of activated complements in the stratum corneum. Now, these activated complements are highly chemotaxic, and they can induce migration of neutrophils to the stratum corneum. So in the psoriatic skin and also in the IMQ-treated skin, the stratum corneum actually has living cells, and this is the neutrophils that is migrating into the stratum corneum. And these migrating neutrophils form what we call Monroe microabscess. The Monroe microabscess contains neutrophils, and this is a very characteristic feature of psoriatic skin and also IMQ-treated skin. Now, incidentally, Monroe microabscess is named after an Australian dermatologist in the 1900s. Now, so it's observed that when you activate neutrophils, you can induce the secretion of IL-17. And it's now recognized that neutrophils are a major source of IL-17 in psoriasis. So how does exosomes inhibit or reduce IL-17? So we propose that the exosomes penetrate the stratum corneum and inhibit complement activation. So with the inhibition of complement activation, we can lead to inhibition of IL-17 secretion and reduce IL-17. Now, during our in my lab, when we were looking at this hypothesis, we were quite curious as to how potent is this interplay of complements and neutrophils and IL-17 in driving inflammation. And the last year has probably indicated to us that this is a very potent interplay, because if you have been following the COVID-19 situation, you realize that complements, neutrophils, and IL-17 has been implicated in COVID respiratory failure. So we think that this hypothesis is well placed to explain why exosomes could be therapeutic. Well, topically applied exosomes could be therapeutic for the treatment of psoriasis. So in conclusion, we think that topical application for MSC exosomes could potentially reduce the IL-23, IL-17 axis in psoriasis. So with this, I'd like to thank you for your attention and also the contributors to this work. Thank you very much.
Thank you, Sai Kiang, for your overview of the work that you've been doing on exosomes in psoriasis and in other applications. Just thank you. We'll move over to the questions now. We have a couple in the chat box.
Yes.
The first one is again from Professor Kuldeep Sidhu, and he's asking if you would mind elaborating on your exosome purification protocol.
Yes, yeah, we have actually described our protocol many times since 2010. It's based on size, so so we use a filter to filter out the size. But as you can imagine, this is not exosome purification process per se. Is we would like to call it an enrichment process. And size exclusion is widely used, or size fractionation is widely used in exosome preparation now. So you can, in fact, the myself 2018 has described the various purification protocol in great detail, and the pros and cons of using each of these protocol. It depends ultimately on your end use for the preparation. So whether you want to use it for diagnostic purposes or therapeutic purposes or just basic biological research, I think there is pros and cons with each purification protocol.
Thank you.
Yes.
There's a couple of questions here, and I'm just trying to work out which the order to. I should have mentioned before that if the participants would like to upvote questions, you can do that as well, so that we know which questions have the most interest in case we are running a little short on time to answer them. But I will go to the next question here, and that is from Prem Kumar, who's asking, because your MSCs secrete a variety of paracrine factors such as cytokines, growth factors, microvesicles, and exosomes, the combined activity of the secretome has the immunomodulatory, angiogenic, wound healing effects, and they're wondering how can exosomes alone exert the same effects, and if you have any way that you'd like to elaborate on that.
Yeah, so so a lot of the cytokines growth factors are actually present in the so-called exosome preparation. So in any exosome preparation, some of them are inside the exosome, some are outside, and some we do not know if they are inside or outside. But I think at the end of the day is we need to look at the exosome preparation as a whole. Thus, in a particular condition, does it work for us? So all these factors will work in some kind of synergy. The final synergistic state will depend on the disease. And the reason I say that is because if you look at a lot of the proteins that we identify as being therapeutics are actually enzymes. So enzymes is most of the time dependent on the microenvironment. So the equilibrium actually shifts according to the relative ratio of products to substrate. So for example, CD59. So if the complements are not activated, CD59 is inert. When you have OECD cannot work unless you have a research much on the environment. So I like to think of exosome as being some kind of smart drug because it works according to the environment.
Thank you.
So I'm not sure if I answered that question correct. It's a bit more complicated. And in fact, I have a paper published a couple of years ago where I advocate the proteins as being the functional component of and why it works to proteins. And in that, I also elaborate a little bit on the enzyme activity of the exosome.
Okay, thank you. We've just got time for one more question now, and then perhaps we can come. Okay. Thank you very much. So I just was a little disconcerted there because the recording stopped and then started again. But
yeah,
I
so
think
I'm
we're
not
back
sure.
on. We're back on track there. Just thank you for your presentation. We'll now hand over to Anne from the group. He has been again very involved with exosomes over a long period of time. In one of the revision paper in the Journal of Cellular Vesicles on how to characterize exosomes, and he's going to talk to us a little bit about his research work in the neurodegenerative important aspects of having these positions, characterization and comparability of these. So
cool. Well, thank you, Janet. Thank you, Janet, for that introduction, and thank you to the organisers for giving me the opportunity to speak to you today. And I've been asked to speak on two topics. One is about our work looking at the role of exosomes and extracellular vesicles in neurodegenerative diseases, but also to talk about standardisation in EV research, standardisation and rigour. So I'm from La Trobe University, and I'm in the La Trobe Institute for Molecular Science, which is in this very nice building here. And we have a large group of researchers working on extracellular vesicles, and we have a research centre based around extracellular vesicles. So I'll tell you a bit about the work we're doing. Sorry, my screen. Here we go. So to start with, the field of extracellular vesicles and exosomes is in one way an old field. It's been known that cells release vesicles into the extracellular environment since the 1960s, when particles released by platelets were known as platelet dust. But since the early 1980s, when the term exosome was first coined, and this was to describe vesicles, small 100 nanometer sized vesicles released by cells. And this was in work looking at the transfer of the transferrin receptor, that the field has really started to grow. And these are just four graphs from various papers published over the last few years that just show you some of the growth in the field. And one thing to start talking about is one issue that the field has at the moment, which is around nomenclature. So the term extracellular vesicle captures all kinds of vesicles that are released by cells. And exosomes are a subset of extracellular vesicles. You've probably heard of other types of EV, such as microvesicles, ectosomes, prostosomes, and oncosomes. And so this first graph here, which was published in 2014, shows the growth of the term microvesicle and exosome. And we can see over the last few years that there's been a rapid adoption of the term extracellular vesicle, which again captures all those different kinds of vesicles. There's obviously been a large growth in the amount of research grants on EV research, which is great to see. And also, particularly since 2010, a rapid increase in the number of patents that have been awarded for extracellular vesicle research. So this is demonstrating that this field of research is rapidly entering the translation phase. I think one pivotal point over the last sort of 15 years is a paper from 2017, there were two papers, 2007, 2008, sorry, that showed that extracellular vesicles can contain nucleic acid materials such as RNA, and this has really spurned a rapid adoption of looking at EVs for their potential as diagnostics, which is something I'll touch on at the end of my talk. And here's a paper which was led by a previous speaker, Sai-Kiang Lim, which shows the number of PubMed papers, PubMed entries, which have used the terms mesenchymal stem or stromal cells and either exosomes, EVs in the title. And again, you can see a very rapid increase in the number of publications in PubMed containing those search terms. So this is a very rapid growing field at the moment. So with a rapidly growing field, it's also spurned the formation of an international society, which we've heard about through the last two talks. And the International Society for Extracellular Vesicles was first formed in 2011. This was after a really pivotal meeting that was held in Paris. And currently there are more than 1,500 members of the society in 2020. And the society has been really important, as I'll show you in the next few slides, in sort of capturing the community's involvement in EV research and starting to produce some guidelines, such as the MySEF guidelines you've heard about, and also position papers and developing a rigor and standardization group, which I'll also tell you about. Other key things that ICEF have been involved in is obviously developing annual meetings, also workshops. There are several workshops that have been held on very specific topics to do with exosomes and EVs, and also running scientific seminars. When the society was formed in 2011, a journal was established, the Journal of Extracellular Vesicles. So this is about 10 years old now, and it has a very healthy impact factor. It's an open access journal, and it's really got some very nice position papers and the MySEF guidelines, which are all readily accessible, and I'll be happy to share links with anyone who's interested in those papers. The Society is also heavily involved in developing educational resources. We launched a massive open online course, a MOOC, in 2016 through the Coursera platform. This was revised in 2019, and there's a current revision of that course, which is being produced at the moment. So if you really want to get a bachelor in EV research, you can go and do this freely available MOOC course. And I'm going to talk really today about the standardization and rigor efforts driven by the Society, and also the some of the position papers that have been produced. And you can get a lot of information about the Society from the website icef.org. So one example of a position paper that's been produced by ICEF in collaboration with the International Society for Cell and Gene Therapy was this one that was published last year on providing a statement on the use of EVs from mesenchymal stromal cells and other cells, and considerations for their potential as therapeutic agents for treating COVID-19. And so, again, this is something where the Society, as it's grown and the work has started to move more translational, we've been reaching out to related societies such as ISCT to develop these position papers. So the first guidelines for EV research were published in 2014. And the executive board of the Society sat down, and we came up with these minimal experimental guidelines, which were then termed MySEF. So MySEF stands for Minimal Information for Studies of EVs. And this early paper really captured, at the time, what were the best guidelines for undertaking EV research. And some of the things that we came up with in this paper was that any EV study must characterize as much as possible the EV population analysed, and we've heard through the two talks today about the heterogeneity of vesicles in a particular population, and also to demonstrate the association of any functional feature attributed to the EVs or exosomes by their specific co-isolation. And following publication of those MySEF 2014 guidelines, another thing that ICEF does is it reaches out to the community to ask their opinion on things. So we reached out and tried to measure the impact of those MySEF 2014 guidelines. And we found that a significant number of researchers who publish on EVs had read the MySEV guidelines, and about 3/4 in their EV publications had followed and cited the MySEV 2014 guidelines. We also asked the question, are minimal requirements needed? And there's always one person who says no, but pretty much the majority of individuals answering the survey agreed that minimal requirements were needed. So this spurred us to move on with developing what's now known as MySEV 2018. And instead of the executive board of the society coming up with these guidelines, this was a real community-based effort. It was led by Clotilde Thery, who's now the president of ICEV, and Ken Whitwer, who's the executive chair of Science and Meetings for ICEV. And this involved reaching out to EV researchers and inviting their feedback on several different aspects of EV and exosome research. And by doing this, we collated the general feelings about the various topics, and this produced those 2018 guidelines. And it covered such topics as nomenclature, also the collection and pre-processing of samples, including pre-analytical variables that need to be considered, especially for doing diagnostic or therapeutic production of EVs. Also, there's a whole section on EV separation and concentration, characterization, functional studies, and also some general considerations around reporting requirements, telling people what you've actually done so that those experiments could hopefully be reproduced in another laboratory. And also mentioning when there are some exceptions, because this is a really new growing field, so there will be some exceptions to these guidelines. And so I've just put a little note here that these guidelines are exactly that, they're guidelines. So they're meant to guide and improve the field, and these guidelines are going to evolve with time as we get more technical advances in analysing EVs and from what we understand about their biology, therapeutic and diagnostic applications. And currently there's work going on to produce the next version of these guidelines, which we hope will be published next year. This is just the masthead of that paper. So there were over 380 EV researchers that contributed to this manuscript, and it was published in 2018. And I've already put a link in the chat for you to go and have a look at it. It is quite an in-depth document. So if you're new to the field, it might seem quite confronting to look at all this detail, but there's a really helpful checklist at the end of the paper that you can use to see what are the major features, what's the major information you need for performing EV studies. I'm also going to highlight another aspect of rigor and standardization in EV research, and that is a website called EV Track, which was produced by Arne Hendricks, who collaborated with several EV researchers to generate this really nice online tool. And what this online tool does is you can send your manuscript to EV Track, and you'll get scored as to how well you have reported your experimental conditions, reported on the separation methods you've used, the kind of markers you've used, how you've analysed your exosomes, counting them and sizing them. And there's more than nearly 1,700 publications now in the EV Track database, covering more than 3,200 experiments. So it's a great resource. And what you get when you've submitted your paper to this database is you get a score of how well your paper has reported on the methodologies for doing EV research, matching things such as the MySEV guidelines. And we can see that there's been a real great increase in the scores that papers are receiving. So the average score in 2015 was about 22%, and now in 2021, the average EV metric score is just over 60%, showing that there's been a real adoption of transparent reporting and standardization of methods in EV research. In terms of rigor and standardization, ICEV's also established a committee, and this is aimed at reproducibility and comparability of different studies. And part of this is to avoid things such as interpreting results. And again, coming back to that, are given properties or functions really due to EVs themselves rather than co-isolated components? Or is it a specific subtype of EVs, such as exosomes, which are a small subset of the whole range of EVs that can be released by cells, really displaying specific or unique functions as composed to other EVs such as microvesicles and microparticles. The subcommittee of ICEV, the Rigor and Standardisation Subcommittee, has been going for a couple of years now. It's been led by Rink Newland and Juan Manuel Falcon Perez, and they've set up several task forces, and these include a blood EV roadmap, one on reference materials. Both of these have published manuscripts in the Journal of Extracellular Vesicles recently. There's, of interest to this group, there's a task force on unproven therapies and regulatory affairs, which is quite active at the moment. There's one on urinary EVs, isolating them from urine, cerebral spinal fluid, and also bacterial EVs. And if you'd like more information on these task forces, you can go to the ICEV website here and get that information. There's also an editorial, again published in JEV, which explains the setup of this committee and also how you can become involved. So now I'm going to completely switch gears and talk a little bit about the work that we do in my lab, where we've been looking at the role of EVs in neurodegenerative diseases. And we've been doing this for about 15 years now. And the diseases we're looking at include things such as Alzheimer's disease and Parkinson's disease, as well as a group of diseases called prion diseases, which include things such as Creutzfeldt-Jakob disease, CJD, and bovine spongiform encephalopathy or mad cow disease. And a common feature of these brain diseases is that they occur in the aging population, and they're all associated with proteins that misfold and deposit in the brain. So we've been looking to see whether there's a role for EVs, given that EVs and exosomes can be released by all different cell types of the brain, are involved in transporting these proteins to different parts of the brain, but also can we use them as biomarkers of disease, because EVs can actually cross the blood-brain barrier and be found in the circulatory system, such as in the blood, and also the cerebral spinal fluid. So we've been analysing the RNA content of exosomes isolated from cell lines, urine, saliva, blood, and as I'll show you, even brain tissue itself. And we can do transcriptomic analysis to measure the expression levels of different microRNAs in the exosomes isolated from in this case, cell cultures. And we found that in a cell culture model of Creutzfeldt-Jakob disease, we could find a panel of 9 microRNAs that had different expression compared to cells that didn't have the disease. So we published this in 2012 and asked the question at the time is, can we use extracellular vesicle microRNA profiles to diagnose neurodegenerative diseases? So diseases of the brain are very hard to diagnose. You need to normally use advanced imaging techniques. So we thought if we could develop, say, a blood test looking at the microRNA profiles in the exosomes or EVs, that might provide a really nice way of diagnosing these disorders. And this is some results from a study we did a few years ago looking at blood-based extracellular vesicle RNA biomarkers for Alzheimer's disease. And what we did was we obtained 23 healthy control participant blood samples and 23 Alzheimer's disease cases. We extracted the EVs from the serum samples, and then we used next generation deep sequencing to profile all the microRNAs we could find in those extracellular vesicles from the serum. We found a panel of 16 microRNAs in those EVs from the serum that were differentially expressed between the Alzheimer's participants and the healthy controls. So this was our discovery phase. We then performed a validation phase where we analysed 50 samples blind to diagnosis using this panel of 16 EV microRNA samples here to see if we could diagnose Alzheimer's disease from healthy controls. And using quantitative real-time PCR on the EV microRNA from the serum samples, we correctly diagnosed 13 out of 15 Alzheimer's patients, giving us a sensitivity of 87%. And we correctly diagnosed 27 patients out of 35 that were healthy controls, giving us a specificity of 77% study. And we found that 3 of these patients here in the healthy controls actually had what we think might be preclinical Alzheimer's disease. And if we find that they have traversed into developing Alzheimer's disease, that might raise our specificity up to about 91%. So we think that analysing EVs in serum particularly their microRNA, can be a really useful diagnostic, and this is being used for many other diseases such as cancer, other infectious diseases as well. I mentioned also that we've been working on techniques of isolating extracellular vesicles from tissue samples, and here I'm showing some EVs that we've isolated from frozen human brain samples, and this has allowed us to look in more depth at the role of these EVs play in these neurodegenerative diseases. It also allows us to compare the RNA profile that's found inside these little vesicles from the brain with those we find in the blood as well. And we've done this also in animal studies, and here we have an animal study of an infectious neurodegenerative disease called Creutzfeldt-Jakob disease, where we have an animal model that we can inject with the infectious agent, the prion that causes these diseases, and follow longitudinally the microRNA expression profile in exosomes from blood samples of these mice. And what we found, and this is work we recently published, we found that we can find a whole lot of preclinical microRNA markers that show up before any clinical signs of the disease are found, as well as clinical microRNA biomarkers. So this is just highlighting that we can do these longitudinal studies and that we are picking up some really relevant biomarkers for a disease that could hopefully diagnose it before the clinical symptoms appear. And in that mouse study, we identified a panel of 14 EV microRNAs that gave us a discrimination between healthy controls and those with the prion disease. And we applied this in a sample of human samples, blood samples that we obtained from Germany. We used those microRNAs we found in the mice, and doing this in the human samples, we found that we could discriminate the healthy controls from the CJD patients with quite good sensitivity and specificity. So EV microRNAs are a promising tool for the diagnostics of several neurodegenerative diseases, and as I've mentioned, can also be applied to cancers and infectious disease. And finally, I'd just like to acknowledge the many people that are involved in the work that we've been doing on exosomes and EVs over many years. And thank you very much for listening, and I'm happy to take any questions.
Thank you very much, Andy, for covering so much ground in such a short period of time. It's great to have the summary of all the work that's been going on in the International Society for Extracellular Vesicles, and to also touch on the fact that we are not just looking at EVs as potential therapeutics, but the fantastic and diagnostic opportunities that they present. We've got a couple of questions that have come through specifically for you. We have one here from Kaldip Sidhu to ask a question around what sort of impact have the Society of EVs guidelines had on the regulatory framework that's being developed in the US through the FDA, and more specifically locally here with the TGA. And perhaps cynically, Kaldip saying probably none so far, and do you want to speculate on why? And perhaps some of the others that have participated in these guidelines might also like to comment here.
So the guidelines, have mainly covered experimental work on EVs. The MICEB 2018 has been cited over 2,500 times already. So we can say in terms of the field of EV research, those guidelines have had a very big impact. In terms of impact on regulatory agencies, I think that's a little bit harder to measure. And that's why ICEV has collaborated with IICT, and perhaps Psychiang could speak to those engagements too. But I think we've recognised the need, as this field that is really, it is still very new and it is rapidly moving into that translational space, that the EV researchers can learn from stem cell and cell therapy researchers about how to move these things into the clinic. So I think we're probably early days with discussions, with the TGA here in Australia, but the rigor and standardisation group does have that task force, which it does include some Australian representation as well. So I think getting that global view into regulatory issues around EV research is really beginning, and it's taken quite a serious step.
Thank Thank you. And there's a question here from Shanti Gurung, who's asking if you could comment on the use of serum or plasma to investigate exosomes in the blood, the benefit of one or the other.
Look, that's a great question. And, it's a question we get asked a lot. We do some studies using plasma. We do some using serum. In this particular, in the Alzheimer's case, we use serum because of the way the samples had been collected. We were trying to use plasma, but for this particular cohort, we were tapping into the Australian Imaging Biomarker and Lifestyle Study of Aging, which had a specific protocol for collecting samples, and that started well before we were doing our EV research. So, that's why we ended up using serum, because we could get reliable results from that, less so from the plasma. I think plasma is probably the preferred blood fraction for doing diagnostic work on. But then again, I think if you're getting a robust biomarker readout in either plasma or serum, I think that's probably good enough.
Thank you. And I'm just going to try and work out which question we should address next. There seems to be quite a number come through, and then we're going to move on to the panel. There was one here from Edna Hardiman. I'm just trying to find that. From Edna Hardiman, and she's asked whether or not there are human conditions or diseases where the amount of EVs will change.
There has been, that's a great question too. And I think there's been some thoughts about EV release increasing, particularly in cancer. Cancer cells are known to be very prolific releases of EVs. So I think in a number of cancers, it's been shown that the EV numbers are actually increased. Counting these very small nanometer-sized vesicles is quite, it's a technical challenge. And so we rely on techniques that count the number of particles in solution. Not all of those are going to be exosomes. So getting to those numerical values is quite difficult. But cancer is one example where more exosomes are released, for instance, in cancer cells than normal cells.
Thanks, Andy. I'd just like to invite Sai Kiang and Rebecca Lim to both turn their cameras back on and join us for our panel. Thank you. We have quite a number of questions, and I'm not sure that we're going to be able to get through them in the remaining 10 minutes. So I have asked that the questions be upvoted. So I'll start with those that have the most votes and throw them over. So Dr. Yagan Bilganti has asked what sort of purity of extracellular vesicles are required for your clinical applications, and then how do you assess that?
Maybe I'll take this question. So I think for MSC exosomes, for clinical indication, it's not so much the purity, but I think it's the reproducibility. So if you have relatively impure, I think you need to reproduce the impurity for every single batch that came off. It's not so much about purity.
I think just on that point, I think that's probably a good lesson that has come across from the cell therapy field, where we originally started thinking that it was just a product was just about what the cells were we thought were in there, but then we realized that the impurities were perhaps as important as the target population. So that's a fascinating comment.
Could I just add, Rebecca highlighted this very nicely in her talk, where she talked about the heterogeneity as well, and finding, I can't remember the term you used back, was it the magic soup? If you think about these vesicles, they're tiny. They cannot contain the full complement of proteins, RNAs, and other molecules in each single vesicle. So it's very difficult to get at sort of potency of a particular population until because you don't know exactly what the mechanism is or what those magic ingredients are in that single vesicle that's having your therapeutic effect. So the idea of purity is you can get a pure population of vesicles, but do you need to go into greater detail than that to get a pure population of active vesicles?
Thank you. So the next question that has a number of votes is on the current purification protocol, and is it suitable to meet therapeutic dose requirements. And I guess that's going to depend on indications, but if we think about something that's not a rare indication, how are we going to get there with the therapeutic dose?
I'll take this question if it's okay.
Yes. Go ahead.
I think the field has been thinking about this. So the efficacy is not very high if you use primary cells. So I think as a whole, various groups are trying to move MSC EV production into the use of clonal immortalized cells, so that we could grow them in large fermenter systems, so that those production is not limiting. And unlike living cells, EVs are a lot easier to produce in large quantity and process in large quantities.
Thank you. I'm not sure if anybody else wants to add to that, or no. Okay, then the next is a comment and a question. It was directed to Andy, and that's regarding your blood EV studies. Is it possible to determine the source of the blood EVs, as in which cells in the blood are they being released from? And by the way, great talk.
Thank you. Thanks for the question. Look, there's been a lot of work trying to identify neuronal markers on EVs that you can, and at the International ICIV meeting a couple of weeks ago, we had this whole session on, there's a controversy around that technique. So it's not really possible yet to really tell where those EVs are coming from, because they're going to come from all over the body. But it'd be really nice to know where your diagnostic marker is coming from, what kind of cell type, because that might give you hints as to how you might treat that disease.
Okay. And a similar question here from Rosa Ignacio, and that is whether if you use serum or plasma, do you get the same mRNA profiles?
We get similar microRNA profiles. So yeah, we are doing some work at the moment to really compare the two. But we've been doing work between cohorts using serum from different cohorts from different countries, and we get very similar results with those.
And a question here from Duncan Thompson. Do you think that EVs you're measuring have pathological effects in driving disease, and therefore are they potential targets for neutralization?
Yeah, we do think the microRNAs we pick up that are dysregulated, when you look at the targets of those, they map back to known disease pathways. So we do think they can be involved in the disease, and also the proteins that are deposited in the brain, they get carried in their pathogenic form on these EVs, so they can act as seeds for that protein misfolding. And it has been shown that if you reduce the number of EVs released by neurons, you can actually slow down some of these diseases, especially in animal models.
Okay, and I think we're getting close to the end of our allotted time, and unfortunately we're not going to be able to answer all of the questions we have, but there's a question here from Daniela, and that is, to what extent do you all predict that the cell-free stem cell derived therapy will become a frontier of cell therapy and diagnostics? And perhaps which diseases are you putting your money on for the first impact? Rebecca.
Oh dear.
We're all working in this space, so you must have some faith, or
yeah, for sure. We've all drunk the Kool-Aid. Totally agree with that. I think from a dose perspective and I guess predictability, so weighing up the risks and benefits, I think the neonatal population has been quite underserved by cell-based therapies because there are real concerns about how an infant who has the rest of their lives ahead of them could potentially, weighing the risks of exposing an infant to that sort of experimental therapy. I'm talking about trials, not an approved therapy, but for trials, I think there's a lot of bandwidth within the neonatal medicine space. They're very small patients. So if you're looking at an IV infusion, the amount of material that needs to be manufactured is far less when you're talking about a 600-gram patient compared to a 60-kilogram patient. And being underserved by cell-based therapies for regen med, if you've got something that will work that is potentially safer, one of the hurdles that we quite often come up against in neonatal medicine with cell-based therapies is, how sure are you that these cells won't mutate, won't turn into a tumor, et cetera, et cetera. When you haven't got a cell and the material that you're administering is static, then you could argue that the risks are therefore lower because there's that element of predictability in there. So for that reason, I would say neonatal medicine would be one of the major ones that would be impacted if we can take this forward.
Yeah, I'd like to add to Rebecca's on the neonatal. In fact, I think the Harvard group of Stella and Alex, I think have started, or they have gotten approval to start a phase 1 clinical trial in their patient, which is the premature babies. I'm not sure if they started yet, but I think they got the FDA approval for a phase 1 study. And also Ava Road in Austria has, I think they have started their phase 1 clinical trial for the use of intracochlear EV implants. And I heard several trials have been approved. So from the regulatory perspective, I would say that EV regulation has started, and I think there's some guidelines on it, although it's confidential, I guess, for every application. But there's definitely a regulatory framework out there already.
Okay. Thank you very much. Unfortunately, I think we're almost out of time, and I do need to do some thank yous. First of all, I'd like to thank all our speakers for participating and engaging in the conversation. There are still a couple of questions left in the Q&A, and if you've got a moment, if you can answer by typing into those before you sign off, that would be appreciated for the participants. Although they may not see them, I'm not sure. I'll get back to you on that one. But also to thank our participants who signed in, joined the call, and to let you know that we do have a poll running, and we'd invite you to take the poll just about how you came to know about the webinar and whether you've taken away any insights and what you'd like to hear next. And of course, I'd like to thank our sponsors, Sartorius, Thermo Fisher Scientific, Cytiva, the Paul Corporation, and Bard One Life Sciences for their support of the meeting. And to say that thank you to New South Wales Stem Cell Network for hosting it, for inviting me to chair the session, putting together the panel. So that's thank you to Professor Bernie Tuck and the New South Wales Stem Cell Network Coordinator, Karen Weiss, who's been sitting in the background helping us. So thank you, everybody, and please take the poll. Thank you, everybody.
Thanks for chairing, Janet.
Thank you.
Thanks, Janet.
Bye.
Bye.